So welcome each and every one of you to this particular course on molecular docking, titled as Advanced Molecular Docking. And my name is Jitesh Doshi. I will be your instructor for this entire course. In this course, let me give you a very quick introduction about what you will be doing, and then I'll ask you some questions so I'll get to know all of you a little bit better. So this course is intended for biologists, biochemists, pharmaceutical chemists, or pharmacy students, and many other related subject students to understand the role of computational techniques in understanding the molecular interactions. That's what the docking is. We'll be going into the details of what the docking is and how do we do that and so on. This is a kind of a workshop based on both theory as well as the demonstration of the software that we'll be using. So you can also perform the hands-on sessions along with whatever material will be given for practice, so that you should get an confidence. What that is the goal at the end of this workshop, to get a confidence in using these tools fluently whenever you need to understand any aspect of the molecular interactions. With this, there are few instructions at the start I would like to give. First of all, I know that many of you may be new to this particular subject, and the subject is very vast. There are a lot of different things that we need to consider. So there are going to be times where you feel that you are not familiar with something, or there are few terminologies on the screen that you don't know, or there might be something that I might just assume that you know, but you have never been studying that because you are all coming from different backgrounds. So it will be really good if you can keep on asking the questions. Ask as many questions as you want, as you can. That will make this session much more interesting, interactive, and fruitful to each and every one of you. At the end of this, you should feel confident about using these tools on your own whenever the need be. The basis for this is going to be some freely accessible open source tools. We're not going to use any commercial or high-fi tools as such. So the basic requirement is that you understand basic of chemistry, basic of biology, and you understand basics of computing, everyday computing, how to edit files, how to save it in different format. For questions, okay, there is a question. Should we unmute our mic or message you? Well, unmuting is really good because you cannot always explain the question in the short message. So you are expected to unmute. So you can just use the option raise hand. As soon as you raise hand, I'll pause so you can unmute yourself and you can ask your question absolutely. Or if you feel that it can be answered later, or it's just something related that you want to put on record and maybe answered later is fine, then you can put it in the message as well. So as soon as I get to that, or I stop talking about what I'm discussing, I will answer that also. Both are absolutely fine. All right. So yeah. So as I mentioned, the goal of this workshop is to get your basic understanding from biological and chemical sciences to the computational sciences and understand how they can be related. Now, I would like to know a little bit about all of you. First of all, so I can already see your names. What would help me articulate this session to be fruitful for all of you is that you let me know your field, what kind of field you're studying, which year you're studying, if you're studying or working, just let me know that you're working as a lecturer, professor, or researcher, whatever it is. And along with that, also give yourself kind of how much understanding you have about molecular docking, so that I know whether I should start from the basics or directly jump into the advanced stuff as it is titled. So you can rate yourself in molecular docking from 1 to 5, 1 being you do not know anything about it, 5 being you have used it at some point in your work or so on. So just kindly do that, each and every one of you. Just give me your subject, year, and your rating in understanding of the doc. So I'll articulate it so that everybody will be uh, able to utilize this platform in a much better capacity. So do that quickly. Fourth year biotech, very new to the docking. That's good. Okay. B Farm, third year, completely new. All right. You don't have to give your names. I can see them already. Final year B Farm. Okay. Again, new to the docking. Pharmaceutical science, okay, in the faculty, okay, that's great. B Farm third year, one, right? New to the subject, BTech, Biotech, rating 3, all right. B Farm final, some understanding, all right. B Farm, okay, all right. That's really good to see that we have a spectrum of audience starting from the earlier years of the B Farm to the master's levels, biotech, and others as well. And most of you, I can see, majority of you are at the beginning of this particular topic. So definitely, I'll be starting with the basics itself, and like we'll talk about each and every aspect of this process so that you understand and don't miss on anything. All right. B Farm third year, okay, mostly B Farm and biotech. So I think the basic background that you need, you already have that. That is basic of chemistry and basic of biology. Both of those will definitely be known to you. So we can start. All right then, guys. So let's start our today's introductory session. Today is just the first day, so we will mostly talk about some basic things that we need to set up the foundation of molecular docking. We'll get a quick introduction, and then we'll move on to some of the ways by which we can use these type of techniques in variety of different ways. Some applications we'll talk about, some limitations we'll talk about, and then we'll talk about some practical aspect of performing the molecular docking. So again, I welcome all of you to this particular session on introduction to the molecular docking. And let's start. What is the basis of molecular docking? First of all, what does it do, or why do we need this kind of a technique in the first place? So let me tell you, whenever in our normal studies, experimental analysis, whenever we need to understand a certain biochemical process or a biochemical reaction, whether it has happened or not, we use variety of biochemical reactions, as you know, and they are manifested in variety of different ways to be able to understood what is causing those to or whether the reaction is happening or not. For example, here are three different, just some of the examples. So we have here a test which is called as a catalyst test. So this test is usually done to understand the presence of certain microorganisms which contain the enzyme catalase in them. The test is very simple. You take your microbial culture, you mix it with the hydrogen peroxide. If the catalase enzyme is present, this enzyme converts the hydrogen peroxide into water and oxygen. Oxygen is released as a gas, and this causes the effervescence or bubbles. And this is how we know that the test is positive. If it doesn't happen, then we say that the test is negative. There is another test over here where we are performing the target identification by simple microscopic imagery. So in this case, you can see that we are using a very specific inhibitor for a target, and to understand that target is present, we can see the cells before the treatment and after the treatment, and we can see there will be a huge change in the morphology depending on what kind of target has been chosen. So microscopic imagery is another way of identifying these things. Then we have simple reactions like, for example, ligand binding assay, in which we perform the assay or titrate it by different concentrations and mix it with the required reagents. And if the binding happens, then there are some indicators, some markers, which will tell us whether the reaction has happened or not. The color intensity can also tell us sometimes about at what extent the reaction happened. So these are just few. There are many other biochemical reactions that we do on a day-to-day basis to understand whether the molecules that we are expecting, they have interacted with each other or not. Ultimately, no matter how do we manifest these reactions as a macroscopic form, ultimately the basis for each one of this reaction to really happen in nature is exactly the same. That is the molecular interactions or molecular recognition. We'll talk about this process, which is very relevant to the molecular docking. What is happening here in this first case? There is a catalase enzyme which is capable of converting hydrogen peroxide to water and oxygen. So when we put a large amount of this hydrogen peroxide with the culture, the hydrogen peroxide molecules, they will go and bind to the enzyme in their substrate binding site. The enzyme will convert them into or break them into the water and oxygen. That is what the chemical reaction will happen. So the molecular interaction here is very important. If these two molecules are not able to interact with each other, you will not see the result of the test. Similarly here, when you are using a selective inhibitor for a certain target, the molecule goes and inhibits the protein. That means it inhibits the enzyme in its activity. Thus enzyme is no more active, and it causes it to cause a change of physiology, which will result in the change in the morphology of the cell if it is an important part of the cell's morphology. And here also ligand binding, as a for example, what happens when the ligand is binding to a certain target or its receptor, that will release inactivate the protein in some way, or maybe release some product. Ultimately that will be identified by some other indicators, and that will give us this colors. So no matter what we see in the outside, at the basis of this is the molecular interaction. That is the basic chemistry that happens. So the thing is that if we can understand these chemical reactions in their structural form or at the level of the molecular structures, then we will be able to estimate these interactions well before we have to perform these experiments. So the molecular docking works on the principle of the molecular recognition, and that is why we use this. So molecular docking is an kind of alternate way of identifying whether the given two molecules interact with each other or not. And these can be a variety of different types of molecules, but mostly it is used to understand the interaction between the biological macromolecules and the small molecular ligands or other biomolecules. So that is the main domain in which this technique was developed for. So very quickly, let's talk about the molecular recognition process itself, and then we will shift to some basic chemistry, just a quick review. But at any point, if you feel that something is not familiar, let me know. We can discuss that in more details or in an elaborate form, and then only we will go ahead. So as I mentioned, for molecular docking to be understood, we need to understand the underlying process of molecular recognition. As you know, in the nature, in any system which is consisting of molecules, all molecules are always in a constant state of motion, what we call as a Brownian motion. And any cellular process only happens when there is an interaction or a recognition of one molecule by other molecule as its partner. So to be able to have these interactions, what is necessary? Well, to interact with each other, the molecules or the atoms of the molecules, they should apply some forces on each other which are attractive in nature. As we know, the chemical forces, different types of chemical forces are there. They can be attractive, they can be repulsive. The very basic and the simplest force, as for example, we can see the charge dependent forces. As we know, the positive charges will repel each other, while positive negative will interact with each other. So these all interactions that happen at the molecular level, they are governed by variety of intermolecular forces, which and especially of attraction. So only when these two molecules which are coming close to each other, if they have the forces of attraction with each other, then they will come closer, they stay with each other, and that will give them the time and space required to undergo a chemical reaction or a chemical transformation. If the forces interacting of repulsive in nature, they will very quickly go away from each other, and they will try to start looking for some other binding partners in the reaction where they can actually undergo a chemical reaction. So if we can understand the basis of these intermolecular forces of attraction, we will be able to judge these interactions just based on the structure or the atomic arrangements or atomic composition of the molecule. That's the whole idea behind molecular docking. So with this information, the goal of molecular docking studies is to answer two questions. So what we're trying to do with these kind of studies is that, given the structures of two biomolecules, determine if these two molecules can interact with each other favorably. And if they do interact, then answer in what orientation or in what conformation these two maximize the interaction between them and minimize the energy of the entire complex. So the energy is a most important term here, because everything, all of these forces will lead to the energy. This energy will tell us whether the reaction is happening in a favorable manner or unfavorable manner. So all of these basic chemistry that we have talked about, the molecular structures, atomic rearrangements, the conformation, the energy, the forces, everything is interlinked here in molecular docking. And the models which are used for docking, they will kind of come together and give us the view of this. We are not going to directly jump into the techniques of molecular docking. We will set our base. We will understand the foundational principles, that how does it work, how do we understand these structures and so on, and then we will move on to these different practical aspects of it. So this is what we are trying to achieve with molecular docking. Now, to understand the molecular docking, there are few things we need to be aware of, and I will very quickly go over those things so that we will be able to perform this docking in a much appropriate manner. So let's see what are those things are, what we need to know when we are looking for any kind of molecular docking applications, what we are actually getting ourselves into. So to understand molecular docking, which is based on the interactions, these interactions are, I'll just note down some of the key terms here. Wait a second. So we need to understand first the molecular interactions which are present in each. To understand these molecular interactions, we need to be aware of different types of forces which act. So IMF, or intermolecular forces of attraction as well as repulsion. When we know about the forces, to compute those, we need to understand what are the different types of forces which act on each atom. To understand that, we need to understand the structure of the molecule. The structure of the molecule consists of different atoms, different bonds, and with this different conformations that they can have. So these are the key terms of the basis of structure we need to understand to be able to see where it comes from. So based on the atoms, bonds, and conformation, we can understand that what kind of forces they will be able to exert on other molecules or other atoms in the vicinity. And then if we know the forces, we can compute the energy. If we know the energy, we can understand the chemical reaction happening. So that comes from the basic thermodynamics, that how we can understand the energy and the chemical reaction which is happening over there. So our the agenda will be to first understand the molecular structures properly, understand what do they mean and what are the key aspects of the energy, how it is related to the energy, and then going forward, understand the biological systems and their structure and their role. Once we understand that, then we will look at the interaction between these two systems, that how do we generate these kind of models. So molecular docking is a very elaborate technique, and it can be applied to variety of different types of systems. And it can understand variety of different types of interactions which happen in the chemical system. So when we talk that, okay, these interactions are governed by the intermolecular forces or intermolecular interactions, what are these different types of interactions that happen and what is the basis of those? Well, there could be variety of different processes that happen at the chemical level. Some of the most important interactions you might have studied in your basic chemistry is that we have weak van der Waals interactions, which can happen, which are non-bonded in nature and can happen between different atoms. Hydrophobic interactions, when we have any hydrophobic atoms, they can have these interactions. Dipole-dipole interactions, because of the different charge distribution, there is a dipole which shows the direction of the total force which is acting in a molecule, and then it can give us these interactions. Then we have hydrogen bond interactions, which are very important, specifically in biological context, because most of the biomolecules, as you can see, whether it's a protein, it's a DNA, they are held together completely by the hydrogen bonds. They are very strong kind of interactions which stabilize many interactions. So even when the different molecules go and bind to these biological systems, they also form these hydrogen bond interactions with the biological system. So these are also very important. Then we have ion dipole interactions. So different charge ions can interact with the dipole of a certain molecule and interact with it to generate some forces, thus contribution to the energy. Same way we have ion-ion or ionic interactions. and we have covalent interactions, which are the strongest of all of this listed here. So here, as you can see, these are listed in increasing order of their strength. Van der Waals are the weakest, while the covalent are the strongest of the interactions. So covalent interactions, as you know, they are formed when the two atoms, they share their electrons, or their orbitals are overlapping, and that forms an extremely strong connection between those. So these are some of the type of interactions which give rise to a certain chemical reaction that happens. It need not always be the formation or the breaking of covalent bond. It can also be a non-bonded interaction which leads to the certain transformations. So in the most common type of molecular docking, we usually don't talk about the covalent interactions very commonly, but we do talk about rest of the other type of interaction. So given two molecules, we try to understand how many of these attractive interactions are possible between the given two molecules, and based on that, what would be the binding energy of these two systems, so that we can understand its favorability for a chemical transformation or chemical reaction. So these are the things we'll be discussing in this course, that how these are computed, what are the different models, and how do we relate it to the binding of the molecules. In biomolecular systems, the contribution of hydrogen bonds is very important. They are very prevalent, and you can significantly see how they interact with a variety of different systems and compute those also very easily. So what exactly are these interactions? Well, we know about hydrogen bonds. I hope everybody of you know, but still, if you're not aware, those are simplest bond which are caused because of the charge distribution and the partial opposite charges on different molecules. Like even if you look at the simple water molecule or water as such, so you will see that entire water all the water molecules are bound to each other or held together with each other through the series of hydrogen bond interactions. So as you can see here, water molecule has hydrogen, oxygen, hydrogen. Because oxygen being electronegative, it will pull the electrons more towards itself. It will have a partial negative charge. Hydrogens thus will have the partial positive charge. So this positive of this hydrogen will interact with the negative of other oxygen, and this way it will form these bonds. So these kind of bonds are caused as the hydrogen bond. We'll define them very technically later and understand how to compute them all. So a result of a docking is this: once you perform, let's say you have a protein and let's say you have a drug molecule and you want to dock them with each other, what do we get is, with respect to the protein, where exactly is the best position or conformation for this molecule to bind, and what are the different types of interaction that the molecule will have with the protein. That is what one of the type of docking would do for us. This is the kind of output we get, and that's why we do this kind of a docking. So now what we'll do is we'll very quickly talk about the different types of docking, and then we will talk about the basis of molecular structures. That will be our today's agenda. So molecular docking can be done for a variety of molecules. It doesn't have to be always the small molecule and the protein as such, but it can be between variety of different types of molecules. And based on what you're looking for in your system, you can understand these different types of docking. So if you want to understand the interaction between the two different proteins, you can perform the protein-protein docking. In this, the protein molecules, which are very big in size, they are huge, because these kind of interactions are very important in formation of different protein complexes in many chemical reaction, where one protein interacts with another protein, giving rise to the next cascade reaction or activates a cascade reaction. To understand these biomolecular associations, you can use the protein-protein docking as a way to understand how two proteins talk to each other and how do they communicate, what kind of interactions are used, and so on. You can also identify which surface of the protein is involved, what amino acids are part of it, and you can characterize that in those kind of. What is capsid? I didn't say capsid, it's a cascade reaction. Cascade reaction is basically a series of reaction that happens followed one after the other. So when a certain biochemical pathway is activated, it follows a series of reaction. That's what I meant, the cascade reactions that happens. So that is one type of docking that we can perform when your system is a biomolecular system consisting of two or more proteins that can interact with each other. You can also perform the protein peptide docking. Well, proteins and peptides are proteins and peptides are kind of similar type of molecules. Both are made up of amino acids, but the difference is that protein is a complete large folded structure that acts as a functional unit, while peptides are very short stretches of amino acid which may interact with the protein as their ligands, as their modulators, and so on. So to understand the interaction between them, what we do is a protein peptide docking. You can also understand the interaction between the proteins and small molecular systems like drug molecules, inhibitors, modulators, or so many other types of molecules are there for which you want, or there could be chemical probes, there could be metal ions. So you want to understand their interaction with the biomolecular system. You can use the protein ligand docking. This is the most common type of study for which the docking is used to understand the interaction between the small molecule, which could potentially be the useful drugs, and their interaction with the proteins, which are the targets for different diseases. So this is one of the technique we'll be discussing in much more details, but others also we are going to discuss in different capacities. You can also perform the docking between the proteins and DNA. So there are many reactions in which the DNA and proteins are involved. So for example, the DNA replication or polymerization, we have DNA polymerase, which sits on the DNA and binds. We have different proteins like transcription factor binding sites. We have different activator proteins, which different regulator proteins. So these proteins go and bind to the DNA at a certain sequence or a certain region, and then they carry out the chemical reaction that they are supposed to. We have nucleases which can cleave the DNA. We have many kind of these proteins which are very helpful in this. So to understand the chemical reaction happening between the protein and DNA at that point, we can use the protein DNA docking as a way to understand those reactions. Similarly, the way we studied the protein and ligand docking, ligand is any small molecule which goes and bind to a larger macromolecule. So similarly, we can also understand the chemical reactions between the DNA and ligand. So in this case, you can see here a small molecule which is bound to the DNA to perform a certain reaction. So to understand all of these different types, we have different types of molecular docking that has been developed. For each one of this, we have a different software, different tool available to perform these kind of analysis. So essentially what we are doing is, let's say you want to understand the interaction between the two proteins. You understand the role of the protein structures, how they are important. You understand how they can be produced on computer, and how we can use an appropriate program to understand their reactions. That's what we will be doing in this case. Or depending on what system you're using, you can use any one of the docking algorithm that is appropriate. Okay, that's extremely basic of docking, but the technical part is still much more complex. That how exactly does it do all of these things? That will be the discussion for another session. There are also some very specialized types of docking that are there, which we'll be again discussing. Like for example, you can also do the docking with the water molecules around to understand the role of those in the interaction. You can also do the docking to understand the flexibility of the molecule. You might have heard about the INDISFIT model of protein enzyme action. If not, we are going to discuss that. What exactly does it mean? So you can do the docking to understand the flexibility and the role of the rotations of amino acids in their positions to understand the molecular interactions. We can also do the metal ion docking, which are very useful if you want to use some metal complexes as a way, or there are a variety of proteins in our body which contains metal ions in them. To perform their docking, we have very specialized forms of docking. All of these different types ultimately do the same thing. As soon as you represent the molecular structures of your system correctly, you will be able to estimate this interaction by using these tools. So the basic requirement for this is that how are we going to represent a molecular structure on a computer? What are the ways and what information is contained into these systems or these information? So we will be talking about these basic today, what molecular structures are and how to be represent them, and then how we're going to use that. So that's the different types of docking that we can have. Docking can also be done with the help of some chemical flexibility, which can be introduced with the molecules. So based on that, the docking can be called as, you will often find two types of docking. Either it will be a rigid docking. Rigid docking is simply means the molecules that you're using are rigid. So you give a structure of a molecule and exactly as they are, they will be docked. Docking is nothing but finding the best fit. If you want to keep it in the simple terms, finding the best fit between the two molecules, that's what docking means. So rigid docking is the simplest type of docking in which you keep the molecules as rigid just the way they are and just dock it with each other. On the other hand, we also have a flexible docking in which the molecules involved, they are allowed to change their shape. They are allowed to change their conformation. They are allowed to move their atoms around in order to see if there is any other way of this arrangement of these atoms in a structure that can lead to a better energetics or better uh, interactions between the two. So those are also we are going to learn about in this entire course. Now, these are different types of docking. Why do we need the docking? What are the different applications of the molecular docking? What all things we can do with this? We will very quickly go over that. And then let's start about the representation of the molecular structures on computational platform. Molecular docking has been there for some time, and its applications can range in variety of ways in many domains of the biological and biochemical sciences. The basic application I already told you, the goal of the molecular docking is to determine the lowest free energy structure for the receptor and the ligand complex, to understand in which form the two molecules will interact so as to have the lowest energy of binding. The molecular docking can also be used to search the database of molecules and rank them for lead generation. So this application comes from the most important field, which is drug discovery. So to discover the new drug from the molecules which are already available in the databases, we can simply apply this application or this software to find out which molecule has the best possibility of being a lead in our drug discovery study. So that can be used. We can use the molecular docking to calculate properties like differential binding of the ligand to two different macromolecular receptors. This is again a very important step when you want to find out. Let's say, for example, one of the application of this is side effect. As we know, many drugs that we have in our daily usage or we use in our day-to-day life, most of them have some or the other side effects. And many of these side effects are caused because of the cross reactivity of the molecule. That means because the molecule is able to interact with more than one specified targets. It may interact with some other important parts of our body, other important proteins in our body, and that's what might cause some of the side effects. So if you want to study the role of these molecules in potential side effects, you can use the molecular docking as a way to investigate these interactions, to identify what other molecules from the human proteome this molecule will be able to interact with favorably. So that will give you a chance of identifying potential problems in the molecule that you have designed as a drug, just as an example. But there are again many things we can do with this. You can study at the geometric level the structural details of this and understand what kind of geometry is important for the molecule to have an activity. Or you can study the normal protein and its ligand or receptor and its stimulant or enzyme and its substrate. These kind of complexes you can study geometrically, the molecular geometry of course, and understand what kind of geometry is important for these kind of interactions. The molecular docking can essentially be used in enhancing the utility of the small molecules which are potential drug candidates. So for example, to propose the modifications in a molecule and find out what more can be done to make the molecule interact in a better way with the target, we can use that. This process is called as optimization process. I don't think we will be talking much about it in this course. We don't have that much of time, but it can be used. The application of docking also comes when you want to generate a very new molecule, a novel molecule, and test its efficacy before you go to the experimental part on a computer itself. So we can use in de novo design of different leads. Leads are nothing but the compounds which are potentially be successful in the drug discovery projects and so on. A library is just a library of, actually it's not the library design, rather we can use it for library screening, so that if you have a library of compounds that you want to screen for the better activity, you can screen them. So these are some of the areas in which the docking can be very helpful. There are many applications of docking, and there are many systems which have been studied very well with the help of molecular docking. There are many drugs which are come into the market with the molecular docking studies in initial role, so that you can use it in variety of different projects. So with this, I'm going to talk more about molecular docking, but later. Before that, I wanted to make sure that we understand the molecular structures properly, and then we will talk about what kind of software will help us in each one of these studies. All right, let's go to the next presentation. All right. So as I mentioned, to actually perform the docking, you need to first understand the basis, the molecular structures themselves, how they are created, what is the role of each one of the information in the molecular docking. So our next topic of discussion is going to be the representation of molecular structures in molecular docking studies. Okay, so we learn about first how they are represented, and then how do we use this information. Molecular structures are the key ingredient in any one of these studies. What exactly is a molecular structure? Well, molecular structure depicts a very specific arrangement of atoms in a given molecule. As we know, every molecule is made up of different types of atoms. Every molecule is represented in a certain geometric form, because that's how the chemistry happens. We may talk about this chemistry a little bit in detail. So structure of a molecule is nothing but a specific arrangement of atoms in a three-dimensional space. So for example, when we look at the water molecule, we write it as chemically H2O. We know that it contains 2 hydrogens and 1 oxygen. But in three-dimensional space, when these atoms are present in reality, the oxygen is present and it is connected to hydrogens at a certain angle. So it forms a chemical structure in which there is a certain distance between the atom of oxygen and hydrogen, what we call as a bond length. A certain bond length will be there. Then these hydrogens are directed in such a way that they form a certain angle between these two bonds, what we call as a bond angle. So to define a molecular structure of water, we will need 3 things: the definition of all the atoms involved, the bond lengths between every pair of connected atoms, and the third is the bond angle between every pair of bonds which is present, consecutive bonds which is present. So these 3 things can help us draw the structure. But in this case, the let's say, for example, we have another molecule, let's say H2O2. Now in this case, how exactly the structure is? So for example, we have an oxygen attached, and let's say we have hydrogen attached. Okay, for example. Now, this is a very simplistic linear representation, but what are the bond angles? What are the bond lengths that will determine how they are arranged? But in this case, now that we have 4 atoms, only having these 3 components is not enough. We also want to identify something that is in 3D space, because angles are 2D geometry. They only can tell us in 2-dimensional plane how the things are placed. But if you want to find out in 3D, like for example, if this H is behind the plane or in front of the plane, or it is above the plane or below the plane, to define these 3D geometries, we need a 4th coordinate in this. That is what we call as a, let's say some atom is there, we call as a torsional angle. So that is a third type of coordinate we need. So torsional angle, just for those who are again new to this term, or it's also can be complementary with the dihedral angle. So when 4 atoms are connected to each other, the first 3 atoms, they can create one geometric plane, 2D. Next 3 atoms can have another geometric plane, and the angle between them is called as torsional angle. Essentially, what it shows is that in 3D, what are the positions of each one of the atom, and that's what we need. So for any molecular structure to be defined, we need these components. Very quickly, I'll go over this again, because I'm not sure how many of you have studied all of these in detail. So when you want to study the molecular structure, it's defined by the bond lengths and bond angles. Then, I mean, those are the four things that I talked about, and these are the few properties which we associate with the every molecular structure. It tells us about all of these properties. They ultimately will, based on the type of atoms present, their charges, we can understand the bond polarity, which also is very important. And in three-dimensional, they can attain different configurations or different conformations based on the torsional angles that we talked about. So when you want to define a molecule, you want to say that this is the structure of a molecule, you need to give all of these different things associated with the structure. Only then you can say that's a complete molecular structure. Now, why do we have these different angles, different lengths, different polarities? Well, that is because of the chemical theory that you might have already studied, that how do the atoms are made up of, what kind of electron orbitals they have, how do they hybridize with each other, how do they cause the repulsions, electron repulsions, and this form a very certain structure. So that discussion, I don't think I should do it right now. I hope all of you are familiar with that. So why do we need to worry about this detailed representation of a structure when we report the molecular structure on a computer? The reason is that very simple. These structures or these arrangements are the key to understand their function. These are the ones which are responsible for any property or any function of the molecule that they do, whether how do they interact, what function do they do. Everything will be dependent on what kind of structure do they have. If their structure changes, the function is also going to change. So even if you take a simple example, like different kind of chemical isomers, let's say enantiomers. So if the structure in an enantiomer into is a slightly different, just around one atom, but the properties that they possess are completely different from each other. Or you take about the cis trans conformations, again they might have a different properties with when it comes to the binding to certain molecules and so on. So these structures can help us understand the functions very well, and that's why we need to get to the details of this structure when we represent this data. Traditionally, you already might have seen variety of chemical structures which have been represented by simple 2D diagrams, like for something like, let's say, benzene is represented like this, or we have simple cyclohexene, which is represented like this. But our world is not 2D. The molecules do not exist as a 2D molecule, but rather they have their arrangements made in a 3D, and that's what we are expected to find for every molecule. Now, in reality, how does this come into picture? How does the 3D structure is formed? It's very simple. There is a very strong correlation between the structure of or arrangement of the atoms and the energy of the atoms, which causes these kind of things. So that's the reason why we need to learn about these structures very well before we use them in any of the application. Let's take another example. Let's say a simple structure of methane, for example. As we know, we express the methane on a paper or in any document, something like this. We know, or CH4 simply. We know that it is made up of 1 carbon and 4 hydrogen. But then what is the structure or arrangement of these atoms in actual 3D coordinate system? That is what the real structure of the methane is. So what happens in this case? Carbon has these 4 valence electrons, which are going to share themselves with the hydrogen electrons, which only have single electron. There is going to be the electron overlapping in the orbitals, and that's what will cause this covalent bond to form. But because these orbitals are not like this line, they have different types of shape. We have spherical shape, we have dumbbell shape, and so we have these different kinds of orbital shapes that we have done. So because of this, there is a electron repulsion that happens, and it pushes these atoms away from each other. Now, because of this push, because of these forces being introduced, there will be an increase in the energy of the system, and the molecule will be forced to stabilize itself at the lower energy where these repulsion is the minimal or no repulsion. And that's why when we see the 3D structure of the methane, there is a carbon, there is a hydrogen, there is a hydrogen here, another hydrogen here, and another hydrogen behind the plane, as if you can imagine. So this kind of a tetrahedral shape is what is formed for the methane, and that is what the 3D structure of methane looks like. Now, this is a very small molecule, so it's easy to understand and interpret this and know it. But for all the molecules, we cannot understand this right away just by looking at their molecular formula. We had to have some method of representing these chemical structures in their correct 3D form so as to make better use of them in any computation. If these structures are not correctly represented, for example, by mistake, you use a methane structure where the hydrogens are attached in this way in 3D haphazardly, the properties, the binding of this that you're going to see is going to be completely unreal. It will not show the real interaction that are happening between the molecules. So that's the reason these molecules needs to be very correctly represented before you use them for any kind of molecular docking protocols, so that you don't miss out any important information. So based on these arrangements, there are different protocols have been developed which can find out the most stable or the minimum energy structure of any given molecule. So no, for example, when you have the cyclohexane, it doesn't stay as this. It has either, as you might have studied, chair conformation or a board conformation. That is the 3D structure in which it is found. Because whatever interaction happened within the atoms of carbon and hydrogens of this, because of that. Benzene being a double bond, they are anyways rigid, so we don't have any rotations around them. These atoms cannot really move, they are restricted. So it is always present as a planar molecule. It doesn't change its 3D geometry. So hydrogens, of course, can be there in 3D, but overall, the plane of the ring remains the constant. Now, as and when you go on increasing the size of these molecules, you will see that the different types of structures are possible. One molecule can exist in multiple possible conformations, but it chooses to be available only in very few conformations. Again, the reason is the energy. If it kind of be in any other conformation, it is going to destabilize it, increase the energy, which is not important. So it will stabilize itself at a certain equilibrium position of the atoms and then cause the lowest energy or more stability. So the aim of the molecular docking representation is to represent these molecular structures in their most stable conformation to be used for any kind of computation that we do. Remember that if the structural representation is wrong for your molecules, then we will be having very wrong results in the docking. So just for example, let's say we have a protein, we have amino acid which is bound here. Let's say amino acid has a carboxylic group over here in this way. Okay, let's say it is in the negatively charged state at this moment at a certain pH. So when a small molecule that we are going to have, so if we lock it in a wrong representation or a wrong conformation, what will happen is that it will not be able to interact with these atoms correctly. But it may be some other, let's say we have a simple atom, nitrobenzene, we can. Okay, let's just have a very simple atom. So let's see, we have carbon, we have another one, and we have hydrogens over there, for example. So if you don't represent it a correct 3D, when it goes and tries to bind itself in this, it is going to have a wrong conformation. So the interaction that will be interpreted will be wrong. Now, obviously, we do allow it to have complete flexibility, but there are a variety of problems that can arise, or it may take a longer time to find its conformation, which is not energy minimal. And that is the reason when you are trying to dock these molecules with each other, it's expected that you have the best possible representation of these molecules, which can be, of course, changing from here onwards, but it has to be that. So because of that, these representation matter. How do you represent your molecular structure? How do we compute its 3D structure or geometry? That is all very important. And that's where the topic of molecular representation is very important. So how do we represent these molecules and what actually they kind of show us, what information do they provide us when we use these molecular structures for docking? Before that, is this part, I mean, I know that it's just kind of a revision of everything that has been studied in basic chemistry, but still any point that you are not yet compatible with, let me know. Have you understood why do we need to have a correct representation at least of the molecular structures when we represent them on a computer? This is going to be more complex when it comes to the proteins. Okay. Okay, the question is docking is done between the two things. By that, how can we know the structure of one molecule? Well, for performing the docking, we have to first know the structures of both the entities involved. Whether you want to dock the protein or you want to dock the protein and the ligand, you first have to know the structures of those both. Only then you can perform the docking. From docking, you will not know the structure later. You have to know it before you perform the docking. We'll see where do we get these structures, how do we know the structure for the known molecular entities and so on. We'll talk about some of the databases from where we can get these structures downloaded. So that's where the representation is the key to achieve a better results in the molecular docking. Now 2D structures you already have seen many times, but as I mentioned, 3D structures are more relevant for molecular docking because they represent the most close to reality arrangement of the atoms of all the molecules involved. The 2D structures, why we use them? Usually we use them because they are extremely easy to manipulate, very easy to do the calculations on, but they are far from reality. So these are the different levels of representation of molecular structures. First is a simple nomenclature. So we can represent it by certain name. In this case, phenylalanine. We can represent them 2D structure, which depicts the atoms involved and the connectivity between the atoms. So by looking at this, as you have been trained to understand what this representation means, you know that here we have carbon, oxygen, oxygen, hydrogen. We have NH2. These are also carbons, as we know. These double lines show the double bonds. So they only show the connectivity and the types of atoms which are present in a given molecule. Apart from that, there are going to be, of course, hydrogens which are implicitly considered. We'll talk about them as well. But in realistic way, this even though these can exist in multiple, so for example, this COOH can be in this direction, this can be in this direction, it can be in this, it can be in this, but in reality we find it only in one of the way. The reason is again those repulsions and the energies which are present. So this, what conformation does it present in reality? That is what we represent in a 3D structure. It tells us every atom's position in the three-dimensional geometric platform, and it tells us what will be the conformation that we are looking for. This is just again a simplified representation of the molecule. We know that these stick means that there is a bond. These tips of the sticks means that there are the atoms. But a more realistic view of looking at the molecular structures is by using the surface representation. What the surface representation does, rather than showing this in a cartoonish or some simplified way, it creates a molecular surface. As we know, every atom is going to have the electrons orbiting around it, a cloud of electrons. They are going to overlap. So this overlapping causes of, as you can see, a sphere of influence, as you can imagine. And that is what the real structure looks like of a molecule, because that entire area, even though it may have a vacuum for large space, it still is under the influence of all the subatomic particles which are present. So the molecular surface tries to represent that entire view of the molecule, which makes it easy to visualize what kind of interactions are happening, whether there will be an repulsion between the atoms and so on. So all of this is represented by this. So these are the different levels at which we can represent the molecules on a computer based on our requirement. For biomolecules like proteins and DNA, the 2D structures are not possible because they are extremely big structures. It's very difficult to lay them down flat as a 2D structure. So often they need to be represented in 3D surface, 3D structures or surface representation only. So that is how we represent the structures. But how this information is given on a computer to the software? How does the computer read this information or data about the structure of a molecule? So this information is provided to the computer or a software in a form of coordinate system or a form of coordinates. Again, as I'm assuming here, most of you are familiar with what a coordinate system is. Coordinate system is a geometric system to place any entity in a 2D or 3D space or even multidimensional space in different ways. So we usually are familiar with our XY coordinate system, which used for plotting the data and all, which shows us that the position of any or any one of the entity or a data point in X and Y coordinate. But our universe, as I mentioned, is 3D. So we also have a third coordinate, which is called as Z coordinate. It explains the position of each atom in this particular 3D coordinate system. So if let's say we have a structure something like this, then the this particular atom is present in some 3D space, certain distance in negative x-axis, certain distance in this y-axis, and certain distance in z-axis. So all of these three values, they represent the position of this atom. And similarly, every atom is represented as a molecular structure in terms of its molecular coordinate or the coordinates of their positions. And that's how we represent this molecular structures. So these are a variety of ways through which we can represent the molecular structures on a computer. But for the sake of this particular study, we are only going to make use of the connection tables. We are not going to look at any other molecules representation as of now. So now here we have a certain molecule. Can someone identify what molecule is this on the top right corner? Does anyone know what kind of molecule is this? Anybody? Acetic anhydride. Okay. All right. Aspirin, right? It is the structure of aspirin or acetyl salicylic acid. That's what this structure belongs to. So one of you has answered correctly. Now we know this because we have learned about this. We know what it means. We know that when we see the structure like this, it means that we have this carbon atom here. We have another carbon. This carbon is connected to three other hydrogens because the valency of the carbon is 4. That's why we need to fulfill that, and so on. We know that there is a carbon CH over here. Here also we have CH, and so on. So all of this we know because we have learned about it. So the same aspirin molecule, when you want to represent its structure on a computer or in a computational file format, how does it use this data? How the molecular docking software understand all of these positions of the molecular structure? So for that, the most common way of representing this entire structure to a computer is by using something that we call as a connection table. Connection table is a table of all the atoms listed one by one and their connections and their positioning. So this is how a connection table looks like. So whenever you download the structure of any molecule from the internet, from the databases, or even if you draw it yourself, ultimately it will be saved as these kind of a data. This is one of the type of data. So what does it represent? As you can see, it's just some list of all the atoms present. So every line in this file, it corresponds to individual atom which is present. So the first line usually states like, which algorithm or which software has generated this file. The next line is a simple identification line which shows how many total number of atoms are there in this molecule, how many total number of bonds are there in this molecule. Then the next few lines, they represent the atoms. Every atom is represented on a single line. So I'll show you what information is there. So this is the first atom. So the first value is the x coordinate that we can draw. The second value is y coordinate, and the third column is a z coordinate. In this case, the z coordinate values are all 0. That means it is a 2D structure. But if it is a 3D structure, you will also have the values which are for z coordinate. The next column represents what type of atom it is. So here you can see at this position, that is -3 and -1, -3 and -1, we have a carbon atom. So that is what it shows us. The second is -3, -2, which is also a carbon. So another carbon is present at this location. So if you follow these coordinates, you will actually be able to draw the entire molecular structure, which is represented just by this structure. So this is how the data is represented by giving the coordinates of all the atoms. Like, as you can see, we have carbons, oxygens, and so on. One thing you will observe